Hello, everybody. My name is Ivan Kirov. I'm an associate professor of radiology and neurology at New York University Long Health. My topic for ENIGMA-U is MR spectroscopy. Sharing the screen now. Go to slide number 1, and let's start with a basic introduction to MR spectroscopy of the brain. I will cover concepts in this first lecture. And the most basic concept of all is how MRS, which is short for MR spectroscopy, is different from MRI. The MRI image that you see on the left is actually a map of the hydrogens of the water molecule. Because there is so much water in the brain, this map looks like a picture. Of course, the brain is not just water, and MR spectroscopy gives us a way to look at other constituents of the brain. And there's different types of MRS that allow us to do that. With proton MRS, we can detect protons of molecules other than water. And there's phosphorus MRS, carbon MRS, and so on. And here are some examples. With proton MRS, here I'm showing the molecule N-acetylaspartate, which is detected by detecting its methyl group that is highlighted. Phosphorus MRS can detect a variety of phosphorus-containing compounds, for example, phosphocreatine. We can look at water also by detecting its oxygen using oxygen MRS. There is also fluorine MRS, but there is no fluorine in the body that we can detect with MRI. So fluorine MRS is usually used to track delivery of drugs because many drugs do have fluorine. So the different types of MRS allow us to detect different molecules other than water in the brain. And all these molecules have very important information. We can study the chemical makeup of the body. We can look at rates of chemical processes, equilibrium of ions, and so forth. So MRS is a very exciting field. I will only talk about, however, proton MRS, which is the most common type of MRS, the easiest one to do, because it does not require any additional hardware beyond what is already available for doing a conventional MRI. In fact, most scanners have MRS sequences ready to be used, and MRS is used in the clinic as well. Okay, so let's take a look at MRS of the brain. On the right-hand side, this is called a spectrum. This is the result of a proton MRS experiment of the brain. You would see two peaks. The largest one belongs to the hydrogens of the water. The second largest peak is that of the subcutaneous lipids. But we don't really need those peaks. We are interested in the area between them, which contains much smaller peaks. This is called a spectrum, or spectra in plural. How do we get from the spectrum with the large water peak to the spectrum of interest underneath? We need to get rid of this water. We do water suppression. That is done using a variety of ways on the level of revising the MRS sequence. We need to avoid the lipids. And here is where I introduce the concept of a voxel. A voxel is a three-dimensional pixel from where we acquire the signal. So if we place this voxel anywhere not close to the lipids, we just avoid them. This is the usual way of doing it. There's other ways as well. Okay, what can we expect to see on this spectrum? What kind of molecules? They need to be small. They need to be present in large amounts, relatively large amounts. They need to be mobile, and the protons that are detected should not be exchangeable with the surrounding water. when we apply this criteria, we end up with a short list of about 4 or 5 metabolites that we can see. These are the most commonly measured ones. Another 5 or 6 can also be obtained with more complicated experiments. Let's concentrate on those metabolites, the 4 most common ones that form the well-recognized spectrum of the brain, an example of which is shown at the bottom. The largest peak in this spectrum belongs to the hydrogens, to the methyl group shown here of N-acetylspartate, the molecule that I already showed, and you will hear about in more detail later. We have creatine here. This peak is the sum of creatine and phosphocreatine. The peak next to it is composed of the methyl groups of choline-containing compounds. And the smallest peak on the left of the choline peak is that of the myo-inositol. We'll go over these metabolites in detail in a moment. Before that, I wanted just to tell you about the chemical shift effect, which forms the basic of the MRS. So no MRS lecture will be complete without it. In short, different molecules can be distinguished based on the frequencies at which their nuclei resonate. What does this mean? It means that every atom in every molecule experiences a different molecular environment. It's nestled between different atoms. So the hydrogens that we detect here, they each experience different molecular environment because of the different electron clouds on them. And that means that they also see different degree of the main magnetic field, which is generated by the MRI magnet. The MRI machine generates this magnetic field, and depending on how much of an electron cloud each of those detectable proton has on it, that determines how much of the magnetic field the proton sees, so to speak. And that's called shielding. The bigger the electron cloud, the more shielded the proton, and vice versa. You can see the most shielded of these 4 metabolites is NAA, the least shielded is myo-inositol. So they're separated on the spectrum. They're separated by different frequencies. The units here, the horizontal scale is frequency, and the units are parts per million. Okay, this is a very basic way of understanding this, but it's very fundamental to MRS. What can the brain metabolites tell us? Why are we interested in measuring them? They can tell us about what is going on in the brain, and specifically in the different cell types of the brain. The brain is composed of neurons and glia, the glial cells being astrocytes and oligodendrocytes. And 2 of these metabolites that we can detect with proton MRS are markers for a certain cell type. NAA is a marker for neurons. It reflects neuronal health. Here you can see on the cartoon on the right-hand side a neuron which is the same color as the NAA. And you can see next to it the supporting cell in green, that is the astrocyte. The myo-inositol signal is thought to come from astrocytes. So myo-inositol is known as an astrocytic marker. So here we have 2 metabolites that are markers for 2 very important cell types of the central nervous system, and that's really, really important. The choline peak reflects the degree of cell membrane turnover. It reflects the presence of molecules that are needed to maintain and build cell walls. While creatine reflects cellular energy, because phosphocreatine, which is part of this peak, is a main energy source for the brain. So in sum, important aspects of brain biochemistry are reflected by these 4 markers, and the most major one of which is neuronal health, which can be inferred by looking at NAA. So how are these MRS metabolites used? This is very important. the area under each peak is proportional to the amount of metabolite. So measuring the peak area will allow us to determine how much of the metabolite there is. And that quantity can be either normal or abnormal. And that result tells us if a certain aspect of tissue health is impaired. Here I'm showing an example of choline. On the right-hand side is a spectrum with normal-looking choline, meaning that the peak in proportion to the other peaks looks like it's a normal at normal levels. On the left-hand side, the choline peak is higher, which means there is more choline there. This high amount of choline can be interpreted in different ways, but that means there is something going on with the membrane turnover. And here is a summary of how we use these metabolites. Here, the main examples which are related to important pathophysiology. Low NAA reflects neuronal dysfunction or neuronal loss. It's very important to know if any of the two is happening. High myoinositol may reflect more astrocytes, which is known as astrogliosis, if they're arranged in a certain way, or it can mean high rates of astrocytic metabolism. High choline reflects breakdown or buildup of cell membranes that is abnormal. So, for example, if a lot of cells are broken down by an infection, for example, choline may increase. If there's more cells being made, means that there's more cell walls being made, and of course, cancer is the prime example of this. High creatine reflects higher cell count most likely. Usually the creatine peak does not change as much as the other peaks, but certainly it can change, and it may reflect the presence of more cells than usual, such as in, for example, astrogliosis or cancer. This is just a summary which I won't go over, but you can pause and take a look at. It refers to how we can interpret different changes in these peaks. If these metabolites increase, which means that their peaks would get larger, or they would get smaller based on a comparison with some standard. But in any case, what I want to say here, which is quite important, is that changes in the peaks should not be viewed in isolation. We cannot see a spectrum and know what's going on in this tissue. There's always a context. The context means why is the patient there? What is the hypothesis? What are we expecting to see? So we should always have this information in mind when interpreting changes in a spectrum. MRS is used in the clinic mostly in two ways, and I'm talking about the brain, but also about the body as well. In a differential diagnosis setting, which the most common example is the following. A patient undergoes a conventional MRI exam, and there is a finding. There is a spot on the brain that appears abnormal, and the clinician and the radiologist are unsure of what that is. And they may have two or more possibilities that they're entertaining, but they're not sure what is the case. So they may order MRS if it's known that the spectrum can distinguish between these two or more conditions. This is called differential diagnosis, to see which diagnosis is the real one. MRS can also be used in cancer patients because it can differentiate between dead tissue, which can occur as a result of chemotherapy or radiotherapy, rather, and active tumor. And in research, MRS is used very widely. And the goal, well, one of the goals is to be able to bring some of these research applications to the clinic, but also not necessarily. MRS can be used on its own to study basic biology, basic pathways, basic disease mechanisms. And with this, I will conclude this first introductory lecture. And here is a list of references that I'm recommending to you for a more in-depth look at MRS. There's 4 journal articles and a textbook that I wholeheartedly recommend. Okay, we'll continue in the next lecture.